Hello everyone, so today will be a long video, so instead of doing a live stream and then editing it later, what I'm going to do is I'm just going to pre-record this and upload it all edited so it doesn't take as long as the actual procedure. So what I'm doing today is I'm actually starting the hormone experiment with the algae, and I've taken the algae out, as you can see here, and I'm going to mix it around a little just to make sure it's all mixed around. Essentially what I'm going to do is I'm going to take 10 million cells out of here and then transfer them into each of these beakers, and then what's left here I'm going to reduce to 10 million as well, and then I'm going to use that to start the experiment, and then I'm going to bring it back up to 500 milliliters using this nutrient solution. But to do that I first have to count how many are in here, and the way I'm going to do it is I'm going to take a sample of 200 milliliters and put it into this tube. I'm going to place that there, and now I'm going to take my hemocytometer. I'm going to load the algae onto the hemocytometer. Okay, now that I've loaded that up on there, I'll put it onto the microscope, and there are my cells. And if you haven't seen my video on how to count, do cell counts with a hemocytometer, check that video out. But essentially, a hemocytometer is a tool to count cells or organisms under a microscope. And I got a count of 51. Now the equation is 51 times 10,000 divided by 8 times 500. And we're at 31.8 million. Now, in order to figure out how many milliliters of this solution I need to transfer 10 million trichomes, I essentially have to take the trichomes per milliliter, that number. I take 10 million divided by that number. Now, luckily, this right here is the trichomes per milliliter, so I can simply just use this number right here. So I need to transfer 157 milliliters of the solution. Now it's always a good idea to make sure to mix this again, just to make sure nothing settles at the bottom. Now normally I don't measure things using beakers, however I've already pre-measured this using distilled water to ensure that it's exactly 157. That way I can fill it up to that line and then I can pour it into each. And that should be 10 million right there, give or take. Obviously, there's always some kind of error with measurements, but that's where the important thing is to understand where your errors are. That way you can report them properly when you report your findings. It's impossible to not have any errors. You just have to understand them and understand whether those errors are enough to make your results significant or insignificant. Now what I'm going to do is I'm actually going to remove the rest to bring this down to 157. I need to take out 29 milliliters. And this time to do that I will use this syringe here. So all of them should have 10 million trichomes. So now what I have to do is I have lines tell me that it's 500. And what I'm going to do is I'm going to fill these back up to 500. And this is the growth media that I have. Look at that, just enough. And once I transfer it to the incubator, I'll add the hormones. Another thing I'm going to do is I'm going to take and I'm going to add this to these little centrifuge tubes. And then I'm going to put them in the freezer. It's going to be like a stock solution that I'll save for another time, essentially. And then what I'll do is I'll join you in the incubator cam to watch the rest of it. Hello everyone, so now that I've gotten the boring moving the algae around stuff out of the way, now I'm going to actually add the hormones. And the way this experiment will work is I'll be adding it every 4 days, starting today, for 60 days, and then every 4 days I'll be taking measurements of each of the population of each container. And then I'll also be taking some pictures to see if there's any morphological changes, like essentially change the shape of the algae. Now I keep the hormones in this little case so the light doesn't interfere with it. Indole acetic acid will be first. Okay, I gotta set it to 30 microliters. So that's the indole acetic acid. And next is the control. And what I do is I aspirate the pipette, which is essentially a fancy way of saying I move it like this a couple times. That way it mixes thoroughly, and I can get as much of the indole acetic acid in there as possible. Or in this case, the control. Now finally I need the methylene acetic acid. Now that's the final of the hormones. And now the first measurement I'll take will be actually 24 hours, and then I'll be doing measurements every 4 days for 60 days. And pretty much that's it for now. I'll check in again in 24 hours with this. Make sure to like and subscribe and all that good stuff, and see you next time.